Hi, my name is Mohuli Ganguly. I am in the 12th grade and I go to LaSalle High School in Pasadena. Today, I will be talking to you about my research in understanding the impacts of COVID-19 spike proteins on stem cell-derived cardiomyocytes. This was observable by protein markers staining for changes in cell morphology. The problem that my research was understanding was how does the COVID-19 spike protein affect the cell morphology of HIPSC-derived cardiomyocytes? And I hypothesized that if cardiomyocytes derived from HIPSCs are treated with the COVID-19 spike protein, then the cell morphology of the cells will change and have negative effects for the cellular impulses. My protocols were as follows: stem cell growth procedure, cell plating, HIPSC to cardiomyocyte differentiation, a transfection procedure for the COVID-19 spike protein, and an antibody staining procedure with two different antibodies, NKX25 and TNNT2. My results showed that the control group had optimal growth. The cell yield was as appropriate, and it showed no excessive staining of the NKX 2.5 and TNNT2, which indicates that these cells were healthy and grown to the normal without any kind of exposure to anything. So they were the control group. Next, I had a 3.7 microliter lipofectamine treated transfected COVID-19 exposed cell group. As you can see, it had a little bit more color to it. So in this next slide, it's going to show you the TNT2, which is just one of the antibodies, and the cell clusters that are in the 3.7 and the 7.5 are much more prominent here and are signaling that the TNNT2 is active in these locations, which is very valuable information. And again, as you can see, the NKX25 was again signaled here, while in the control group, it was very non-fluorescent. There was very limited amounts of fluorescence. You couldn't see the stain as prominently as you can in both the 3.7 and 7.5 microliter. And then this is the DAPI staining. As you can see, the cells had several healthy nuclei in the control group, while it had virtually no healthy nuclei in the 3.7 microliter group, with a few cell clusters in the 7.5. This is very important to note, these large cell clusters in the 7.5, as they indicate a type of cell formation called syncytia, which is when there are cell clusters with several nuclei. So these are the cell clusters here. And as you can see by the bright field and the DAPI both match in terms of cell clusters and structures. These larger clusters that you can see in the bright field for the 7.5 are marked in the DAPI as well, which show that these are the live nuclei in these clusters. So as I said, there were no changes in the cell morphology for the control group. They had good growth and virtually lacked any kind of presence of antibody staining for both the antibodies. Then for the 3.7 lipofectamine group, they had adequate staining, basically no DAPI, very limited DAPI nuclei staining, and they had a bit of clustering, but not as much as the 7.5, where there was bright fluorescent staining of the antibodies with clear specific clusters, and also minimal cell growth. So like I was talking about earlier with the syncytia, which is basically when you have cell formations and clusters of many cells cluster and form together as a result of some kind of external stimuli. In this case, I would think it was the COVID-19 exposed cells, as this was not seen in the control group. The antibody staining NKX25 is to indicates impairment of electrical activity of cells, and in previous research that I have read, there have been obvious electrophysiological differences between healthy cardiomyocytes and COVID-exposed cardiomyocytes, and those impairments could be seen with the antibody staining that indicated this. And then for the TNT2, it was a gene that releases a type of protein called troponin, which is used which is released when there is damage to cells, to heart cells specifically. And because this TNT2 was so active, it shows that these cells may have undergone some sort of damage as they were exposed to the COVID-19 um, uh, spike protein. And lastly, the TNT2 also indicates alteration in the calcium ions and ion concentrations, which is an important factor in cell growth and cell maintenance, and may explain why these cells were not able to grow at the same level as the control group. So those were my results. Thank you so much for listening.